I really believe bioprocess development starts with understanding the cell biology at scale. Cells are social entities and they sense each other, they remodel their microenvironment, they rewire their signaling cascade when things such as cell density, mass transfer, mechanical cues change. When we are moving from the T flask to the bioreactor, I think we are not just increasing volume, we are changing the entire context in which the biology of the cells happens. So that's why I think that we really need to respect the biology as a primary design input.
Imagine a world where we could predict how your body would respond to a therapy before it ever enters clinical trials. Today's guest, Catarina Brito from ITQB NOVA and iBET in Portugal, is making that vision tangible. She's pioneering 3D cell models that recreate the complex microenvironment of human tissues, from brain to liver to tumors, revolutionizing how we test biologics and gene therapies. If you're tired of the limitations of petri dishes and animal models, this conversation will reshape how you think about preclinical research. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Catarina, welcome to the Smart Biotech Scientist. It's good to have you on today.
Hi, David. Many thanks for having me here. I'm glad to be talking.
It's a pleasure. Catarina, share something that you believe about bioprocess development that most people disagree with.
Well, I think people are starting to believe it in the bioprocess industry, maybe not as much as I do from the beginning. It's really a strong conviction of mine because I really believe bioprocess development starts with understanding the cell biology at scale. Cells are social entities and they sense each other, they remodel their microenvironment, they rewire their signaling cascade when things such as cell density, mass transfer, mechanical cues change. When we are moving from the T flask to the bioreactor, I think we are not just increasing volume, we are changing the entire context in which the biology of the cells happens. So that's why I think that we really need to respect the biology as a primary design input. So media, oxygenation delivery, to keep biology in the right operation window, so that we can do scale up, thinking about more than just equipment and scale, but really starting with biology as the foundation.
Scale up is much more than quote unquote scaling up. And you said it very well. There's so many different parts you have to look at when you're scaling up a process. Before we dive into our topic today, let's talk about yourself, Catarina. Draw us into your story. Tell us what sparked your interest into biotechnology and how did you arrive in this very exciting field you're now researching in?
Okay. It all started during my PhD. It was leveraged by the questions I was trying to answer. So I was studying mechanisms driven by glycan-protein interactions. And these mechanisms are very different between murine cells and human cells. So that was early on like a wake-up call. It made me really think about human biology and accuracy of the models we were trying to use and the needs that was at the time even higher of models that really reflected. And the cellular processes I was studying, it was neural cells, axonal outgrowth. So these are processes that are very dependent on the context, on the matrix. So we were growing neurons on these murine neurons on these plastic surfaces that were so artificial compared to what's happening in the brain. So this notion of the need for better models of the physical context and how it shapes signaling and morphology and neuronal connectivity that is important. And then, of course, the cellular context, because neurons are not in isolation, and a lot of these cues are coming from the cells that are neighboring cells, that are glia. So the models we were using were monocultures, was also an aspect quite frustrating. And all this led me to try to find a postdoc in which I could tackle some of these questions. My motivation for all this truly crystallized when I had the opportunity to join iBET for my postdoc. I had the chance to work under Paula Alves' supervision. And indeed, she has done at the time already a very pioneering work on 3D culture systems and on showing that it's possible to have biology as design input for models. It was also a very exciting time. It was the first time we were using human pluripotent stem cells in Portugal. So really this experience was very important and really set the direction for my career in trying to build models that integrate human multicellular complexity.
Yeah, that's definitely exciting. Let's start by unpacking this because not everyone listening today is familiar with animal models, with 3D culture, with 2D culture. There's a lot going on in this space. So perhaps let's start with the traditional 2D culture and animal models because they have dominated the preclinical research for a long time. What are the critical limitations of these systems and what are the advantages?
They have both. I should start by saying, because we are all I'm always defending advanced models and 3D models, but all models have value, right? And 2D cultures, animal models, they've taught us a lot of biological questions and pathological aspects as well. But of course, they also have limitations. And I think that all models have limitations, and understanding the limitations of each are extremely important, especially when we're trying to develop advanced therapies. 2D cultures are cells culture in a flat surface. Typically, they are cultures of immortalized cell lines that can be easily grown. So they give us a lot of control and throughput, but they lack the three-dimensional reality of the tissues. Tissues are not flat. Some tissues are layered, but most tissues are three-dimensional. Cell polarity is altered, diffusion of nutrients, but also of compounds of potential therapeutic is also different, and there is no physical confinement of cells and neighborhood effect between them, so cell-cell interactions also changed a lot. And if we think, for example, of cells in these conditions, their receptor localization, their metabolism, their stress responses, all these change from 2D to 3D or from 2D to tissues. So if we're thinking about development of biologicals, for example, these molecules depend on receptor engagement, on transport, on microenvironment access. So there is a big difference in the biology and in the response to the therapeutics. If we think about the animal models, they are invaluable still because they bring systemic biology. They are a complete organism, but they miss a lot of human-specific aspects from immunology, the glycosylation patterns I mentioned before, a lot of genetic variability, the etiology of the disease. So from false positives and false negatives, let's say, that come from these differences in species that may be screwing the readout. Of course, they continue to be useful, and they are part of a toolbox that should be as complete as possible, but indeed they have their limitations.
There's been quite a push from regulatory agencies, especially from the FDA, to get rid of animal models lately. What is your take? Will we be able to get to a day where we will not use animal models at all? Or is this unrealistic and we will always have a hybrid approach?
So I think we are on a path that may lead to the replacement of animals, although a lot of validation is still required because there's a lot of efforts to develop multi-organ systems in which pharmacodynamics, pharmacokinetics can be studied. So far, what has been the push by the regulators is for the types of readout in which we know already we don't need the systemic component. But I would say we are on a path that may go there. We just need strong validation. And there are efforts from the European Commission, inclusively in calls and others, roadmaps to get us there. With also the support towards these aspects of the regulators and of the agencies, we may get there, yes.
This will be a paradigm shift, definitely for the industry. A very different way to develop drugs. And speaking also of a different approach, let's zoom into 3D models and advanced models. Tell us more about that. What is a 3D model and why is it called advanced?
The notion of advance is a bit that it's more than just growing cells in 3 dimensions. So they have to recreate aspects of the tissue they are representing, and this tissue itself, this microenvironment, shapes how cells behave. Is to have a bioactive model that is in which cells are influencing the system, but they are being influenced by the system as well, preferentially in a way in which we attain reproducibility and robustness, let's say. But there are different aspects. There's the architecture, of course, the three-dimensional architecture. As I told you already, important for polarity, the spatial organization, the physical confinement. There's also the important aspect of having diffusion gradients, and these are not just oxygen and nutrients and signaling molecules that are distributed like in the tissues and that influence the biology, but also influence drug penetration, drug binding, and clearance. So these are all important aspects. Then there are mechanical cues and extracellular matrix, which is also important, because our cell sense this stiffness, this tension is also very crucial for survival, migration, for immune evasion, for example, which in cancer therapeutics is also important. And then the multicellular complexity of a tissue, the presence of the different cells. If we think of a tumor, this tumor interacts with stromal cells, with immune cells. If we think of neural tissues, neurons interact with glia and microglia, so these interactions are also important and should be present in these advanced models, so that biology, more closely to what happens in the body, can be recapitulated. And then we can study not only disease, but also predict therapeutic responses.
In those 3D models, Catarina, do people need to visualize that? For instance, if we take one of the cell type, a liver cell type, do we reconstruct a liver organ maybe in a smaller size in that reactor, or do we just have one single cell? I mean, a lot of cells, but just individual cells floating around.
Now we try to, so in trying to recapitulate these tissues, and that's what I meant with this multicellular complexity, is to have precisely the different cell types that compose the tissue interacting between them. And then it depends on the tissue, because like you were saying, in the liver, hepatocytes are well connected between them. They are in very tight junctions. But then you have endothelial cells that have fenestra, that are like windows that allow the passage of compounds. We have macrophages, which are immune cells, which are traveling around, let's say, in the space. So it's trying to recapitulate, if not exactly true architecture of this space, but at least the cell-cell interactions and how they contribute to cell function.
And how long can you keep these cells in the culture? Let's say you want to start a new experiment. Do you have to start from scratch every time? Can you keep somewhat a cell bank? I mean, it's probably the wrong term of it, but you get the picture. How does that work?
So it depends a lot on the cell of origin of the model. So with primary cells, typically they are used and they don't last long. Or they may last in several weeks in the model itself, but they cannot be propagated and proliferated a lot. If we are talking about stem cells, using pluripotent stem cells, then we can differentiate progenitors, at least bank progenitors, and then start already from those. So do it in a multi-step way that facilitates the process. And then the duration of the model itself also depends on the culture system. So we use a lot of bioreactor technology under perfusion. And part of the reason we do it is precisely to be able to prolong the lifespan of these models.
And how do you handle diversity? Because you have liver cells, you have stem cells. I mean, you have all kinds of different cells. Muscle cell, no, probably not muscle cells in your case, but you have different cells. Probably not.
We work mainly in central nervous system, in liver, and in solid tumors.
Exactly, yes. But still, you have quite a diverse cell types, which then have different requirements in a bioreactor setting. So how do you handle that? Are you changing the conditions, the media type, even the size or the shape of your bioreactor? Or how does that work?
The scale, depending on the cell type as well. Each tissue has its own requirements. For example, the neural cells are extremely sensitive to oxygen tension and to mechanical stress, so we use bioreactors, but bioreactor design has to be so that shear stress is minimal compared to others, and we have to have that, and we have to keep low oxygen tensions very constant, so it's really critical to control these process parameters. In the liver, for example, the oxygen tension is completely different, and there it's much more to retain functionality. It's important to have a flow to keep these cells into a rigorous perfusion flow because this contributes to their metabolic competence. For example, to optimize the ratios of cells, because there are different types of cells that we need to put together for the liver models, not just the hepatocytes, but also the so-called non-parenchymal cells that are different types. These ratios of cells have to be very tightly controlled. For example, in the solid tumors, a lot about heterogeneity. It's a lot about having a system in which we can see extracellular matrix remodeling, which is also an important specter of tumors that allow us to create tumor niches, hypoxic niches, for example, which are also important in tumors. And then on top of that, having immune cells and immune cell infiltration. So all these the requirements are much coming from the biology. And then we have to, for each, choose very carefully the variable design so that we can achieve. And then for all of them, as I was mentioning, for us, it's important to have this robustness and this scalability. We try to design models that are as modular as possible and as compatible with bioreactor platforms as possible so that we can sustain reproducibility and throughput. And then in all the systems, we take a lot of care about validations to ascertain the physiological relevance and that it's not lost in scale or throughput.
I'm just curious, I might have missed that, but the purpose of all that is, as you said, is a better reproducibility, but also at the end of the day to accelerate the development. So how do these advanced systems compared, for instance, to an animal model, why can you develop much faster?
The throughput is completely different because if you develop models in these scalable systems, they have an increased throughput and they can be applied earlier in the development. They can be introduced earlier in development of the pharmaceuticals than the animal models, which are usually introduced quite late. So there's a lot of selection that can be done earlier. Also, more and more they are being introduced because a lot of the biology is different. Even in the cancer models, a lot of the new T-cell engagers, multispecific antibodies, they rely on biologies that are not reproduced in animal models. Even if the animal models are available, there's a need to bring human-based models to the front.
We've just scratched the surface of how advanced 3D models are transforming drug development. In part 2, Catarina will dive into the critical role of innate immunity in predicting therapeutic responses and share her vision for AI-powered personalized medicine platforms. If you're finding value in these conversations, please leave a review on Apple Podcasts and your favorite platform. It helps other biotech scientists like you discover these insights. See you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.